next startup team, which was delegated by University of Belgrade, is Perun. We invite the team's first speaker, Petar, to the stage. The floor is yours, Petar.
Hi, can you hear me?
Yes, yeah, perfectly.
And can you see the presentation?
Yes.
Okay, so let's go. We are Perun, and we are making a portable device called AmPipe, which is used as preparation for DNA analysis. Main problem for all our target groups, which are biologists, forensic investigators, and veterinarians, is the inability to conduct the PCR method while on terrain. This means that there's a risk of sample degradation, which can lower the quality of final results or even make the method unusable. Armies around the world are also in need of portable laboratory devices. They're currently examining ways of using portable sequencer on testing the quality of water and soil. AmPlife is a natural fit in this use case because in order to sequence DNA, you first have to amplify it. Another example is that the biggest problem for forensic investigators right now is the inability to conduct DNA analysis on the crime scene. This leaves an open spot for attorneys to dispute the evidence collected, claiming it wasn't handled properly. It is estimated that the laboratory equipment industry will have a 20% increase by 2027. The DNA forensic market will almost triple in value, while the portable diagnostic devices market will have a compound annual growth rate of 10.6%. Why bring terrain to the laboratory when you can bring the laboratory to the terrain? Portability is the key to cost reduction. Our portable thermal cycler device provides user the option of amplifying the wanted DNA sequence on the spot, be it a forest or a rundown motel room. With AmPlife, researchers don't have to worry about sample degradation, preservation, and transportation costs, and what is most important, about quality of the results. AmPlife will have a great impact on researchers' approach to DNA analysis, saving them effort, time, and money. While our competition provides big, cumbersome machines intended to be placed in a room and never moved again, or so-called portable PCR devices which depend on external power supply and logic, AmPlife is a completely standalone portable device. It has its own autonomous power supply and onboard logic, allowing you to change all the parameters without having to hook it up to a computer. Our business model will be B2B and B2G, with the main revenue stream coming from direct device sales and servicing fees. As our main marketing tool, we will be using academic channels, such as publishing work in scientific journals. We need an investment of 200,000 euros for certification, serial production tools, and device production costs. We estimate that we can return that investment by selling around 50 devices. As the number of devices produced increases, the production cost of each device is going to decrease. The biggest leap in this cost reduction is seen when going from 25 to 100 devices produced, and it is a jump from 26 to 45% cost reduction. Our team consists of young enthusiasts which have a great passion to bring something new to the world. We share a vision of accelerating the evolution of laboratory devices towards the completely mobile future. We also enjoy great support ranging from field experts such as Marko Panic, co-founder of Smart Research, and to institutes and government organizations. Next steps for us include finishing the final prototype of the device and, through collaboration with many institutes and faculties that we have a contact with, testing that device in real-world conditions. Join us and amplify life with AmPlife. Thank you for your attention.
Awesome. Thank you, Petar. Are you ready to receive questions from the jury members?
Yeah, we are.
Perfect. Let's do this. Jury, please propose your questions.
Petar, that was a really great presentation and very convincing. So I applaud you. One advice, if you would take from me, is talk a little bit slower so that you make sure everybody understands you, because I think there's none of us here in this meeting who are native speakers. We each speak with our own dialect, or so we each speak English in a different way, and it's sometimes very difficult if you speak very fast to keep track of what you're saying, especially if you're loading so much data and facts on us. And that's really great, but I think sometimes less is more. So make sure that your slides amplify the importance of the key data that you're presenting. On the market side, I remember, I worked at Imperial College, and we had a similar startup, which was called Anywhere HPLC, and they have miniaturized HPLC technology into a handheld device which had a cartridge, a reloadable cartridge. So it worked like Nespresso. The business model was the same, and they could. Their mission was to take the HPLC out onto the field and have people access laboratory technology in rural areas in Africa or South America, where maybe 10 million people had access to one HPLC, and their urine or blood samples would have to wait for weeks or months before they got analyzed. So I see a very similar trend here, and it's a very interesting proposition, but actually quite difficult to how do you cope with the level of precision that is required and that is guaranteed and safeguarded in laboratories which have, as you said, cumbersome and technology or equipment that's meant never to move. So how do you square that circle? How do you make sure that your kit's level of precision and accuracy is comparable to competitors?
So as you can see on this slide, these are the tests done on Center for Human Molecular Genetics at the Faculty of Biology, University of Belgrade. On the left side is a standard machine, Thermo Fisher Biored Proflex PCR system. It is a higher end thermal cycler device. On the right are the results done on our device. The concentration difference between the DNA done on standard PCR devices and on our device is just 8%. And that is taking in fact that we used quite cheap parts for the MVP that we tested.
Did you say 8%?
Yes, 8 to 9%, approximately.
Okay, that sounds really convincing. I'm not sure what I'm seeing. I'm not a molecular biologist, so not sure what I'm seeing makes any sense to me. That would be the other thing, is to make these, tone down these messages and use less jargon in your pitch, so that the average Joe will understand what you're saying.
Okay, thank you. That's a bit that we struggle with, and the speed of my talking.
A question from my side. You mentioned that you have two target markets. One is the B2B, the other the B2G. Yes. Which will be the primary focus group there? As you said, you need to sell approximately 52 devices to be profitable. So which one will be the first one you want to target?
Well, the first one that we want to target is B2B, institutes, faculties, centers that do scientific work and that do have a need for such a portable device, because it is much easier to break into that market segment than, for example, into a military segment market.
Thanks a lot. And the second question to that, if I may follow up with that. So how do you plan to compete with the likes of, for example, Thermo Fisher, who have an army of salespeople and consulting agents getting into these governmental contracts? So how are you planning to crack this nut? Well,
the big benefit on our side is that Thermo Fisher doesn't have a portable thermal cycler device while we do. So, I mean, when they produce one, they will be a direct competition. Until then, they're just an indirect competition to us.
Okay, thanks a lot.
No problem.
Your product sounds like out of the book from The Innovator's Dilemma, where they took hard disk and made it smaller, and established parties couldn't keep up with the new value chain. So I can see that this is a switch for big companies where they won't see the benefit for the type of market that you're addressing in the beginning. My question would be, I was a bit concerned when you started talking about distribution, and that your plan is to publish papers, and that's how you want to get into the the perception of your customers. I'm not sure if this is a good strategy, given like if you see how much a publication is on average viewed by somebody. So what are your kind of thoughts on distributing this to your target audience?
Well, breaking through this target market is quite difficult because it's a niche target market and the regular marketing tricks don't work on them. So our thought process was to lend a few devices to institutes and centers around the Balkans and some in Europe, let them test the device, see how the device works, and all work done on that device, if it should be published in a scientific journal, should cite it that our device was used for it. The other method is just word of mouth, because basically everybody is just buying the standard PCR devices made by Thermo Fisher.
All right. Well, thank you very much.